Some are low-threshold mechanoreceptors that sense touch, others specialize to sense temperature and pain, and still others are proprioceptors, again, responsible for sensing position of the lens. And their specialization continues into the periphery, so the projections that they get the information from are also very specialized, with proprioceptors specifically innervating muscle spindles, and there are many different nerve endings in the skin. And many people have studied the specialization of these skin nerve fibers, David Ginty, for example, at Harvard Medical School. So all of this information comes together not just from the skin and muscle, also from internal organs, such as bladder, highlighted here, and we'll talk a tiny bit about that as well. So my outline today, I'll quickly introduce piezos, tell you about what the protein looks like, its structure, and then mainly summarize the roles of piezos we found in somatosensation and interoception, this sense of internal organs. And then finally, for people who've heard me talk in our board, just a couple of new slides of unpublished data at the end. So the work started with Bertrand Coste, who's a postdoc in my lab, came from Marseille and then went back and runs his lab in Marseille right now. And he came specifically to find mechanosensors in DRGs that are responsible for touch and pain. But after a few months of working, realizing it's too difficult of a problem to find it from DRGs, he decided to take a more reductionist step and try to find it from the cell line. So after, and the assay that he uses, shown in this video, is rather simple. You can see the recording electrode puncturing the cell on my right. And with the glass probe, you can come and poke the cell. And if the cell expresses a mechanically activated current, then you would see, as shown in the bottom, ion channel that's mechanosensitive open and ions come through. So to, again, try to find this receptor in a simpler system, he screened cell lines that are mechanosensitive, and he found that Neuro2A cells that have been used by neuroscientists for decades have a robust mechanically activated current. I won't go through it, but through steps of expression analysis, bioinformatics, etc., he made a list of genes that could potentially be mechanosensor. There are about 300 on this list, and then he proceeded to knock them down by RNA one by one to see which one's responsible for this current. And after 11 months of getting negative data, he finally cloned a gene that was called FAM38A, unknown family 38 member A, that ended up being a mechanosensor that we've been studying for over a decade, that he named piezo1. So piezo1 has a sister, piezo2, at the amino acid level 40% identical, and they have the property that when you overexpress them in a naive cell, you can get mechanically activated currents. And this middle panel illustrates that. The lines above just show that the more you press the cell, the larger inward currents that you see at the bottom. And on the right is just a quantitation of it showing that compared to control naive cells, you can see massive nanoamp currents by simply overexpressing piezo1. So what did piezo1 look like? Most ion channels, of course, have to cross the plasma membrane, and most ion channels have 2 to 6 transmembrane domains. Piezos have 38, which is the most in your genome. On top of that, they come together as a trimer to form this gigantic ion channel in the plasma membrane. Many labs, including McKinnon and Bailong Zhao, solved the structure of cryo-EM, solved the structure of piezo1 using cryo-EM. And as I said, they're trimers. All these ribbons are these transmembrane domains. And at the C-terminus, the 3 proteins come together to form the core structure. The rest of the protein is extended into the plasma membrane into this kind of very unconventional puckering formation. So, as if they're part of a plasma membrane that's being endocytosed. But the hypothesis was, and this was first put forward by Rod McKinnon, is that any tension in the membrane will actually flatten the structures, causing it to open. Hence explaining why they're mechanosensitive. And there is some evidence of this in vitro, both from McKinnon lab as well as by Long Zhao, who is my postdoc and started his own lab in China, have shown that if you actually purify this protein and put somehow mechanical force on it, they will flatten. And this is morphing of the closed ion channels. Pretty remarkable. So just to end the structural things and tell you what we're doing to follow up these studies. So all of these interesting structural findings came from cryo-EM structures, for example, where you pull out the protein from the cell, reconstitute it, and then look at it in vitro. But we're very interested in trying to ask the question of what do piezos look like in native form in the plasma membrane. And the question that we started to ask is, although by less resolution, can we visualize these blades, these three arms, in response to different stimuli by using light microscopy? And so super resolution microscopy has been advancing tremendously over the years, and I just wanted to show you if this is even possible or not. Each circle is the kind of localization principle of different microscopy techniques, and so that's confocal. I'm just realizing this is an older presentation. The newer one is not coming. If someone back there can look and change, it's okay. If not, we'll go with this. So that's the confocal resolution, SIM, STED, STORM. Finally, MinFlux, this newest super resolution microscopy. Stefan Hell devised it, and it's commercially available now, is actually, as you can see, with little piezo trimer there, gives us the resolution that potentially we can look at this. And this is the work of Eric Mulhall in the lab, this Zen picture shows that he's got the mental steadiness to take care of this project. So the experiment is actually quite straightforward. We can transfect cells with a piezo-1 so that we can mark the N-terminus using after transfection click chemistry and put a very small fluorescent probe on it. And we're simply trying to measure the distance between these fluorescent probes in these trimers on fixed tech cells. And so again, just one slide of data, but we were amazed that we can, in these raw renderings, actually see piezo-1 trimers in direct observation of the light microscope. And this kind of summarizes his first set of data, where on the left you can see each dot is the average distance between two blades of one piezo trimer. He's done lots of controls to verify what I'm telling you, but there is interestingly massive variation in the blade distance that we see in hex cells, and the average is actually more extended than what we see in cryo-EM structures. Interestingly, the next panel is that if you treat cells with hypoosmotic shock, which stretches the membrane, and we know electrophysiologically can activate piezo-1, we actually observe the change in the average distance of these interblade distances. And on the right-hand side, you simply see what the maximal distance would be if piezo is completely flattened. So we're seeing about 30% increase on average in channel diameter, which is quite remarkable. What we want to do with these studies is Eric is now actively looking to, instead of one label on each piezo arm, trying to put 6 or 7 of them so that he can have more fine structural data about individual molecules, what they look like, and how does gating can be read out in a native cell, mammalian cell line. Okay, I want to switch now to its physiological roles. And during these last few years, we have used both mouse genetics and clinical data to find out what piezos do in vivo. So piezo-2 was very strongly expressed in dorsal root ganglia, the neurons that I introduced you to initially. And so we made a variety of mouse models to knock out piezo-2 at different times in different places. Piezo-2 null alleles, mice die at birth, so we use various conditional knockouts to address this. So this is just to show that if we knock out piezo-2 from everything down from your lung level posteriorly, we see very, very severe deficits in touch sensation. And this is the typical Bonfrey behavioral assay that you've seen again and again. You're touching the paw with different forces, and in wild-type in gray, you can see the stronger the force, the higher the withdrawal percentage rate of the animals. And you can see how profoundly deficient the piezo-2 knockout animals are. We know this is specific because they respond perfectly fine to temperature, for example. How about proprioception, this sense that I talked about initially? Indeed, without piezo2 mice lose coordination, which is a sign of lack of proprioception. So you can see that these mice are heavily uncoordinated, as illustrating their lack of proprioception. So I'm not showing you too much data, but for each of these behaviors, we can actually, shown below, this is Wilkinson lab, our collaborator, can electrophysiologically show that the deficit is due to lack of mechanotranslection, the initial step of transforming mechanical force into an electrical signal. And this is just single proprioceptors being stretched, and in wild type in black, you see this massive increase in activity that you hardly see in the knockout. One of the very interesting aspects of our study was actually not our work, but the work of Alex Chesler and Karsten Bonneman at the NIH. And they actually identified individuals who lack piezo2. So these are complete null mutations in piezo2. And remarkably, they show the same behaviors and same phenotypes that I described to you in mice, that is complete lack of touch and proprioception. To illustrate the lack of proprioception, Chesler did this experiment where one of these individuals that lacks piezo2 is blindfolded and asked to walk in a straight line. So you and I can do this easily, but you can see this individual struggles. She's harnessed to make sure she doesn't hurt herself. Again, demonstrating lack of proprioception. How about pain? Interestingly, acute pain, mechanical pain, hammer hitting your finger is relatively normal, both in mice and humans without piezo2. But we're also very interested in tactile allodynia, this painful sensation caused by innocuous stimuli like light touch that you and I experience after injury or sunburn, but of course it's a major indication that's folks that suffer from neuropathic pain experience. And we've looked at this both in mice. I'll show first that, so if you inject capsaicin in the hind paw of an animal, you get a very quick, within minutes, this tactile allodynia. In gray, you can see the mechanical threshold of activation gets lowered massively. And in the piezo2 knockout, we don't see this sensitization at all. Again, this is specific because thermal sensitivity occurs normally. So this is in the order of minutes in response to capsaicin. We observe the same thing in response to spared nerve injury and potentially a neuropathic pain model in mice. So as you know, one of the things that we suffer from is that we find lots of targets in mice that end up not translating to humans. But again, here we have the great advantage of these individuals who lack piezo2. And again, this is data from Alex Chesler's lab. So we did a very similar experiment of what I showed you in the mouse, but in humans, imagine two boxes, and these individuals are blindfolded. One box on the arm gets capsaicin and heat, which causes sensitization. The other is the control. You can monitor with laser Doppler imaging that where you applied capsaicin heat, it got inflamed. And then you simply ask these individuals, if you touch A or B, which one is painful? And control individuals, of course, 100% guess that box B was the inflamed part that's painful. And yet the individuals without piezo2 don't feel anything at all. You tell them you have to guess A or B, they guess randomly and get it right 50% of the time. So this is a kind of a powerful, I think, evidence that tactile allodynia depends on piezo2 both in mouse and humans, and at face value suggests that piezo2 might be a good drug target. But there are, of course, complications with this. First of all, given its role in touch and proprioception, I told you, you cannot imagine that you would ever want to take an oral pill that blocks piezo2. The on-site the side effects would be massive. But local kind of applications is a possibility. The other challenge with piezos has been, despite its massive size and facing extracellular side of the cell, there has been very little good pharmacology. And I just wanted to show one slide of our recent published work So we have collaborated with various companies to screen over 2 million compound libraries to find piezo2 agonists and antagonists, and we haven't found any. But recently we revisited, together with Stefano Forli's lab at Scripps, who's designed this Autodoc suite to screen for structural pockets of proteins for chemicals that would bind. And this was a relatively small screen, 100,000 compounds. We're doing this with many, many millions. Found 3 pockets within piezo2 structure from the alpha fold, and just tried to see if we can find compounds that actually modulate piezo. And this virtual screen gave rise to 300 hits that apparently bind piezo2, and when we actually ordered and screened this, we found 10 compounds that seem to be enhancing activity and 6 that are reducing activity, and this is just electrophysiological verification of one of the so-called inhibitors. Again, black is control piezo2 expressed in PEG cells being activated by mechanical force, and in the presence of this compound, a pretty high 100 micromolar, you can see a partial block. Again, on the right, we see the IC50, nothing to write home about, but we're very excited at least to find something and excited to follow this up. So just to kind of put this in summary, piezo2, both in animal models as well as in humans, seems to be responsible for both somatosensation and interoception. This includes touch sensation, proprioception, tactile allodynia, and I need to talk about also mechanical itch. But over the last few years, we've also gotten very interested in this idea of interoception. So these sensory neurons that I talked about, in addition to innervating skin and muscle, they innervate almost every internal organ that we have in our bodies. In addition to the DRGs, there's a whole other sensory system through the vagus, the nodose ganglia, that also innervates these internal organs. And these neurons are gathering information, both mechanical and chemical in nature, and you'll hear more about some of this interoception from David. And they gather this information, but we don't really understand exactly what their roles are in many cases. In some of these cases, the mechanosensation is voluntary, sometimes it's at the unconscious level. So there's a lot to learn. And in collaboration with Steve Liberlis at Harvard, as well as, again, Alex Chester and IAH, we've investigated the role of piezos in various interoceptive roles. And for example, we've shown that piezo2 is required for regulating breathing. And interestingly, not just in mice, individuals without piezo2, the number one reason they go to the hospital is during perinatal times due to difficulty breathing. We've shown that baroreception, which is neuronal mechanism of blood pressure regulation, in mice depends on both piezo1 and 2 together expressed in the nodose ganglia. So these first 2 senses you're not conscious of, but your bladder filling you're very much conscious of, and Kara Marshall and lab showed that piezo2 is required for regulating these reflexes, and once again, in the humans, they don't sense this bladder filling, and so as they grow up, these individuals learn to go to the bathroom 3-4 times a day to avoid accidents. Finally, we just published a story showing that piezo2, again, both in humans and in mice, is required to regulate gut motility. So what I've talked about till now is mainly about neuronal mechanisms of mechanosensation, both in sensory neurons of the dorsal ganglia as well as the nodose. But of course, many cell types experience, sense, and respond to mechanical forces. Blood vessel endothelial cells experience blood flow, muscles get stretched. Many cells, including red blood cells, get indented. And over the years, my lab and many others have studied important roles of piezos, mainly piezo1. So piezo2 mainly expressed in neuron, piezo1 more widely, is involved in a variety of biological processes. So piezo1 is required for lymphatic vessel development. I won't go much into detail. Others have shown important role of piezo1 in bone density, and you might have heard that astronauts who went to outer space come back with reduced bone density. And this is potentially partly through bones actually sensing how much weight bearing they're doing, and this is perhaps partially regulated by piezo. And again, many stories of involvement of piezo-1 in red blood cells and macrophages, etc., that I won't have time to go into today. So for the remaining of the time, I just want to tell you one of these stories of involvement of piezos that ends up being outside of the nervous system. So this is the role of piezos in pregnancy and parturition, and it's work done by Zhao Zhang in the lab. So the reason we started this project is just this intuitive thought that during pregnancy the uterus expands 50 times, and there's got to be mechanosensation involved. And when we looked in the literature, of course, it's known to be very tightly regulated by hormones. There were hints that mechanosensation is important. All the way back in the 60s, people in animal models have put, objects in the uterus to expand it, to put pressure on it. There's no chemicals involved here, and that seemed to be enough to induce the stretch-induced hypertrophy of the uterus, the remodeling that is experienced during pregnancy. And much later, closer during childbirth, just the stretching of, again, in animal models, just stretching of the uterus increases the contractions, again suggesting mechanosensitivity of some sort. When Zhao looked at the expression of piezo-1 and 2 in the uterus, so that it's expressed in both muscle cells, other local cell types, as well as innervating neurons. So when we have a situation like this and we want to find out what piezos are doing, we start by we take the ceiling effect. So we try to delete piezo, both 1 and 2, in every cell type that might be relevant to the uterus. In this case, we use HOXP8, which I've described already before. It's expressed in blue color, you can see, not in the anterior but posterior part of the embryo and then the animal. So then Zhao started looking at both pregnancy and parturition, and by E18.5, this is in mice just before birth, he noticed that the number of pups in gestation were normal, whether in controls or single or double piezo knockout animals, as well as embryo length and weight was normal. So this tells you that, despite what I said in the previous slide, that although mechanosensation has been implicated in progression of pregnancy, at least piezos don't seem to be involved in it. However, when he looked at just a day later, about the time that the pups are delivered in mice, he saw a very strong phenotype only when you lack both piezo-1 and piezo-2. And this just is, on the y-axis, the number of the pups that are retained. Mice usually give rise to 8 to 10 pups at a time. And you can see in the double knockout, some animals end up not delivering 7 pups. In one animal, they were able to deliver all of them. But pretty severe phenotype, given the fact that you almost never see this in control animals. So Zhao wanted to see what the cause of this is, and he used implanted telemetry to measure contractions at various times, as well as combining this with a video recording to see when the pups were being delivered or parturition was happening. So this is one example of intrauterine pressure recordings of a wild-type pup. On the bottom, you can see the light and day cycles, so just exactly at day 19 when the first pup, which is indicated on the graph, shows up, and you can see that's associated with increased intrauterine pressure or contractions. So this is an example of the knockout. This strong upregulation of contractions doesn't happen. So this was one example. This is about 9 control and double knockout animals. You can see pretty consistent phenotype that these larger peak pressures are just not being observed in the knockouts. And you can quantitate this on the right, just measuring median pressure for the wild type and double knockout. You can clearly see the difference. So this lack of contractions, interestingly, are also causing labor deficits and the timing of the birth. So this is again a chart of each row is one female animal, and each blue line is one pup being delivered. And you can see that it's very consistent in the wild type. They all come about the end of day 19, and once one pup comes out, all the rest seem to come up within a an hour or so. This is again very different in the double knockout. You can see that the initiation of the pups coming out varies. It starts anywhere from day 19 to end of 20, and in some of the animals you can observe that the first pup comes almost a whole day before the last one that can be delivered. So this is very consistent with the lack of strong contractions that we observe. And this is where we are. It's a little bit of not complete story, but we're excited about this as an example of how we use piezo's to find novel biology. And the next step is for us, after this ceiling effect, to go in and ask which cell types or neurons are responsible for this phenotype. So instead of knocking out in all cell types now, we go in, for example, and only knock it out in sensory neurons or motor neurons. Interestingly, very preliminarily, both of these are causing no labor phenotype, but when Yunjia injects AAV, so transforming crude recombinase activity, in the uterus itself, is seeing a partial phenotype of this, suggesting that it is a cell type within the uterus that requires piezos to regulate the function that I just described. To summarize this last part, piezos seem to be dispensable for pregnancy but required for labor, and loss of piezo 1 and 2 impairs contractions, which causes this phenotype. And we're again very interested in both finding exactly what cell types are required, as well as how it regulates the lack of strong contractions, which I think would be interesting. In a parting thought, I just wanted to give you how we're thinking about, we've been studying piezos for 14 years now, and I think of this as we're going from phase 1 to phase 2. Phase 2 was kind of checking boxes of saying, yes, piezo does in a system where we knew mechanosensation was very important. And stage 2 is more thinking outside of areas where mechanosensation is very well described, and this molecule helping us find novel biology. And we kind of get into these novel areas through a variety of ways, by expression, because piezos are professional mechanosensors, so wherever we've seen expression, we see an important mechanosensory function. For example, we have a preprint on piezo 2 being expressed in kidney cells that seem to control better reception in the kidney. The whole reason we studied, for example, piezo 2 role in bladder function was from the human clinical data that we knew from our collaboration with Alex, that these individuals couldn't sense their bladder, so that can bring it back to mechanistic studies in mice. And finally, probably my favorite thing is intuition, which is how we started with pregnancy, that there's got to be mechanosensation involved there. Anyway, that's all I had. Just wanted to thank current lab members, alumni, as well as key collaborators for this work. And in blue, I've highlighted, and I mentioned that the people who actually did the work that I presented today. Thank you for